herpes is a sexually transmitted disease which causes a viral skin condition that emerges as cold sores on the lips and the genitals. It's caused by two specific viruses, herpes simplex 1 and herpes simplex 2. The main difference between these viruses is how they're transmitted. Herpes simplex type 1 is transmitted orally, while herpes simplex type 2 is transmitted sexually. Um, Herpes simplex type 1 is a much more common type of herpes. 67% of the global population has this, which is about 3.7 billion people. Whereas herpes simplex type 2, 11% of the global population has it, which is about 417 million people, which is more than the population of the United States. Herpes is a lifelong disease and hides in the neurons of your brain, only coming out in small outbreaks where under like stress or certain emotional conditions, where it will pop up again as sores, painful blisters, and ulcers. So the structure of a herpes virus, first of all, it's in the Herpes viridae family. It's unlike other viruses because it is an envelope virus, so it has a lipid bilayer around it, which is derived from the host cells. On the outside of the envelope are these things called glycoproteins that bind to this lipid membrane and are very distinct and unique to every single virus, and they differentiate the viruses. Inside of the lipid membrane is the pseudomen, which binds to a capsid. This kind of just is a housing for the capsid, which is something that houses the DNA of the virus. The DNA is double-stranded, and the capsid consists of capsomeres in a icosapentahedral shape, and this huge, like weird, crazy, like diamond thing that's pretty whack. And that's the main structure of herpes virus.
Hey, friends! I'm here to talk about the differences between herpes 1 and 2. But before I can talk about the two most common kinds of herpes and their differences, I would like to educate you about all the different kinds of herpes that there are. In general, there are roughly 8 different types of herpes for humans, including herpes simplex virus 1 and 2, the varicella zoster virus, the cytomegalovirus, the Epstein-Barr virus, the human herpes virus 6A, the human herpes virus 6B, the human herpes virus 7, and the Kaposi's sarcoma virus, which is also known as human herpes virus number 8. Occasionally, it's also possible for a simian virus called B virus to infect humans, which is related to the herpes virus, but it's not as common as these types. Now, all of these types of viruses are divided into 3 main groups. Alpha viruses, which have a short replicative cycle and a broad host range, and they're the most common, so herpes simplex 1 and 2 would fall under the alpha type of virus. Beta viruses, which have a long reproductive cycle and a small host range, and examples of viruses that fall under the beta type are the cytomegalovirus virus and the human herpes virus 6 and 7. And the last type of virus that we have available to us is the gamma virus, which is a very restricted host range and includes the Epstein-Barr virus and the human herpes virus 8, and it is the most difficult to diagnose. So now, with this background, we can move on to the specific differences between 1 and 2. So first of all, there are 2 main molecular differences between these two viruses, which there were a few extensive experiments that were done that gave these results. And what scientists found is that both bind to the heparan sulfite glycoprotein C and D, otherwise known as GB and GC homologs. However, it was found out that the herpes simplex virus 1 binds to these far more than the herpes simplex 2 virus does. Specifically, it binds more to, or it has more of these receptors to bind to than herpes simplex virus 1. However, herpes simplex virus 1 is also impacted more by electromagnetic or ionic factors than experimental mutations in the herpes simplex virus 2. which is interesting because all in all, the way that the two are transmitted, and specifically their symptoms, are really not that much different. Herpes simplex virus 1 is the one that is not usually sexually transmitted, and can be found. It involves sores on the lips and face area, and herpes simplex virus 2 usually involves sores around the genitals. However, neither is necessarily restricted to a single area. If you have herpes simplex virus 1, it is possible to get sores on your genitals, and if you have herpes simplex virus 2, it is possible to get sores on your mouth. The important difference is that they are transmitted very differently. Herpes simplex virus 1 is specifically through oral contact, such as sharing toothbrushes, kissing, and eating utensils. And herpes simplex virus 2 is specifically sexual contact that spreads this virus. Oh, and one other thing to mention about the molecular differences between herpes simplex virus 1 and herpes simplex virus 2 is that this heparin sulfate glycoprotein is an important part of the nuclear envelope that mediates binding to the herpes simplex itself. So these things are binding to the herpes simplex virus 1 and herpes simplex virus 2, and the herpes simplex virus 1 has less places for these glycoproteins to bind to, and those binding sites are also more impacted by ionic factors.
Current treatments for herpes include suppressive and episodic treatments, and these both fall under the category of antiviral chemotherapy or antiviral drugs. The terms can be used interchangeably. So both of them use the drugs acyclovir, valacyclovir, and famciclovir. Acyclovir is the oldest drug, so it has the most documented results, and valacyclovir and famciclovir are similar in their effects, but they're newer. So both use the same drugs, but with suppressive, the drugs are taken regularly, and they decrease the frequency of outbreaks, in some cases completely inhibiting them. So it's a lot more effective than the episodic treatment. It can reduce the frequency by 70 to 80% in patients who have really frequent outbreaks, but it also has a higher risk of side effects. Episodic treatment, it's taken irregularly and it decreases the time of outbreaks, but it's a lot less effective, and you have to catch the outbreaks early on or it has almost no effect at all. But it has a lower likelihood of side effects. So let's take a look at those possible side effects. The more common and less dangerous ones are headaches, dizziness, depression, nausea, vomiting, joint pain, skin rash, stuffy nose, and sore throat. Some of the more dangerous, rare ones, but they still exist, are harm to red blood cells, hallucinations, seizure, and problems with speech or vision. So really, there's some potential for a lot of bad stuff if you're taking these. So you always want to consult with a doctor and all that good stuff. For a lot of people, it is just it works to reduce the frequency of outbreaks or reduce the time, and the side effects don't happen and they're totally fine. So yeah, they both control signs and symptoms, and most importantly, they are not a cure. They can't eradicate the virus, and no cure has been found thus far. So these are just the best treatments that we have to suppress the herpes virus.
Which is a pretty important factor of the disease and what makes it so hard to eliminate. So, pretty much all forms of herpes can display this trait. It's not necessarily that every infection will do it, but it's not like only simplex virus 1 or 2 can do it. All of them have this ability. It's just not always constant. This allows it to go dormant and also hide from the immune system. So it can pretty much last in the body when it's not directly causing an infection. And it's not necessarily even reproducing that much either. It's more just staying dormant. This is all facilitated by viral protein 1-2, which doesn't necessarily allow it to, it allows it, so it doesn't have to do with getting into the neuron, but it involves getting to the neuron. So pretty much it allows the herpes to hijack a host cell's dynein motor protein. So So basically, D9 is the same, almost functions like little legs on a cell along a microtubule that would go through the body. And this is like how a lot of transport cells work. So once herpes gets into some kind of transport cell, it would hijack the D9 with the viral protein and basically move it until it encounters a neuron, at which point it would leave the infected cell, probably lysing it and going into the neuron. And then it would remain in there until it starts reproducing and leaves the neuron at some point, as discussed in the section on how herpes leaves remission. So when it's in the nervous system, it can remain in there for years, sometimes without a single outbreak during that entire time, or multiple outbreaks while still keeping some in the nervous system. And at this point, it cannot be easily targeted by T cells because neurons are, for the most part, somewhat protected from the immune system, except in the case of autoimmune diseases, mostly due to the fact that they can't easily reproduce neurons. So it's, they're generally, it's not that they are absolutely immune from the absolutely immune to the T cells, but it's a lot less common that a T cell is going to target a neuron or even look for an infected neuron. And it does still release some copies of itself during this time. It's just not as many, and it's not as aggressive as, say, during an outbreak in the skin. However, traces of herpes have been found in skin cells even during this remission. So it's not necessarily that it is just completely trapped in the immune system and is not affecting anywhere else in your body. It's that it's not having an outbreak anywhere else. And pretty much what this generally means for the disease is that it is almost impossible for your immune system to get rid of it once it's actually established itself. So if you get rid of it early, we'll edit that out. If you get rid of it early, you're good and you won't have herpes for the rest of your life. But once it hits this stage, once it can hijack a cell and start getting into a few neurons, you're pretty much stuck with it for the rest of your life. Though it doesn't necessarily have to have outbreaks.
Alright, so today I'm going to be talking about herpes outbreaks and what causes them and their symptoms and all that jazz. So I'm going to start out with the causes. So scientists don't really know the biological or chemical causes of herpes outbreaks, but we do know a few triggers. So those include physical stress, emotional stress, immune suppression, sex, sweating or irritation around the genitals, drinking alcohol, or certain diets, including nuts and high levels of cholesterol and like that. So all of these things can be triggers for a herpes outbreak. So when you have an outbreak, it usually starts with an initial sexual exposure to the virus, and then 3 to 14 days later, you're going to have some initial symptoms. These are mainly flu-like, and they can be like itching or redness around the genitals, or also just your normal flu symptoms of like a sore throat, coughing, that type of stuff. So for some people, the virus won't really progress past this stage, but for people who have an active outbreak, it will, and those lead to blisters. And in herpes virus 1, those blisters will be around the mouth. In herpes virus 2, they will be around the genitals. And so these blisters will turn into ulcers, which ooze fluid, which is not fun if you have the herpes virus. However, these open sores will crust over and heal eventually, and by about 10 days to 3 weeks, your outbreak should be over. So because of the vast variety of different causes that can lead to an outbreak, and just because of people's different bodies, the frequency of outbreaks really varies, but some generalizations we can make is that over time the frequency and severity of the outbreaks do decrease, and some people can have resistance, which means they will be asymptomatic for the herpes virus. And so that means there can kind of be carriers for it. So even if somebody isn't showing symptoms of the herpes virus, they can still have it. Now going back to what I said earlier about diets and food, so there's been a lot of debate recently about L-lysine and L-arginine, which are two semi-necessary amino acids that in normal healthy people the body creates enough of, and you wouldn't need to take any supplements for that. However, there's been some discussion about the ratio of L-lysine to L-arginine in the body. So back in 1978, there was one study that showed that increased lysine to decreased arginine levels actually prevented herpes outbreaks, and that the opposite promoted them. So that led to a lot of people taking supplements to increase their L-lysine ratios when they thought they were getting symptoms. However, in some recent studies, it's been shown that just taking arginine within 6 hours of having an outbreak helps kill the virus. So that's some other kind of contrasting data. However, even more contemporary studies that went back to this 1978 one just entirely dismissed their findings and said that the ratio of L-lysine to L-arginine has no importance. So obviously there's a lot of varied opinions about this. Basically always consult your doctors before taking supplements, and don't do anything that you just read about online. Always make sure you are doing what's right for you.
There is no cure yet for herpes. Dr. Keith Jerome of Washington University is working on the closest thing we have. By using gene editing, more specifically the CRISPR, which is the most modern and powerful gene editing tool we have, Dr. Jerome is able to target the herpes virus. So Dr. Jerome has created what we call a targeted endonucleus, and by using the CRISPR, he can get the endonucleus to target the herpes virus, which prevents the herpes virus from replicating by inducing multiple mutations, which makes it dormant and unable to replicate.
Herpes is a disease that still affects billions today and is highly stigmatized. And while we're still very far away from a cure, progress is being made every day. So that's our herpes video. Thanks for watching, folks.